Now I'm going to talk about how DNA becomes accessible for gene expression, and we'll focus on the role of acetyl molecules. Again, DNA right here are these little strands wrapped around what's called histones. These histones are just proteins that they just package the DNA inside the cell. They help organize it, and it affects how accessible the DNA is to the gene reading machinery. And then next we have these acetyl molecules. These are small chemical modifications that attach to the histones. And then when the acetyl molecules bind to histones, they cause the DNA to loosen up and become less tightly packed. And once the histone or the acetyl molecules are there, the DNA unwinds as a result and the structure becomes more open. This process is important because it makes the DNA more accessible to the gene reading machinery, allowing it to start the process of gene expression. And since the DNA is unwound, the gene reading machinery can access the DNA more easily. This machinery reads the DNA and creates a mRNA that carries the instructions for protein production. And then when acetyl molecules attach to histones, they trigger a transition from inactive gene to an active gene because the gene is able to be read by the gene reading machinery. And then I again have two questions that I'm supposed to answer from the script. And the first one is, how do acetyl molecules facilitate DNA accessibility? The answer is, acetyl molecules loosen the DNA from the histones, making it easier for the gene reading machinery to access the DNA and begin the process of gene expression. Then number two is, what is the impact on a gene when it becomes accessible? And the answer is, when a gene becomes accessible, it transitions from being inactive to active. The gene can now be read by the gene reading machinery, which leads to the creation of mRNA and ultimately the production of proteins that perform